Bismillahirrahmanirrahim. Biology 9700, A levels, and this is the MCQ paper that we are going to discuss today. This is November 2016, paper 1-1. We start the paper. We have to remember that the syllabus changed in 2016, so this is, of course, on the new syllabus, which, of course, later on was adapted a little later on, but it is valid till 2021. Now, as we approach question 1, the diagram shows a transverse section through a blood capillary. Then there's the wall of the endothelium lumen, and then you've seen this scale bar. Now, this scale bar is very important. This scale bar, it says, is 7 micrometer. Now, what is the magnification of the drawing? Well, all you have to do is measure it. And when you measured it, it was 35 millimeter. So 35 millimeter is equal to 35,000 micrometer, as you can see here. And so 35,000 divided by 7. Please remember the units must be the same. So if this is micrometer, then you have to talk about micrometer. So 35,000 divided by 7, and that is 5,000. And so the answer was D. You must know how to do it. It's not going to be the same question again. So you must know how to do this question. Then we come on to question 2. Question 2 was a culture of human cells had its cell surface membranes removed, releasing the cell contents. This material became contaminated by bacteria. The material was then centrifuged, separating out the various cell structures according to size and mass. Which cell structure would be separated along with the bacteria? Now, the bacteria size, as you can see here, so you've got to see this is that which cell structure would be separated along with the bacteria would be mitochondria. The nucleus is too large and the ribosomes are too small. So that would have been the answer to that. And then this is this diagram, which you will see at the end of this paper again. I'm going to show you this diagram again because so that you can then revise from it. Then coming to question 3, which parts of a cell contain ribosomes? Now, of course, we know chloroplasts have ribosomes, mitochondria have ribosomes, nucleus does not have ribosomes, but which parts of a cell contain ribosome? But then you remember nucleus does not have it, but then inside the nucleus there's a nucleolus. And in the nucleolus, remember, ribosomes are made. Nucleolus is the place where ribosomes are made. In fact, the ribosomal RNA is made from the DNA, which is present in the nucleus, and then the proteins are imported from the cytoplasm because the proteins are made on the ribosome. So nucleolus. So that is why the answer. And 4, cytoplasm has free ribosomes. So we have free ribosomes in the cytoplasm. So ribosomes the site at which all the proteins required by the cell is synthesized. Ribosomes are present in both eukaryotes and prokaryotes, so small that they can only be seen with an electron microscope. The protein synthesized by the cell depends on the specialized function of the cell. For example, red blood cells make hemoglobin, pancreas make insulin, liver cells make protein enzymes like catalase, stomach cells make digestive enzymes, and muscle cells make contractile proteins, actin and myosin. Question 4, the electron micrograph shows parts of 2 eukaryotic cells. And then we had labels W, X, and Y, which of course I've labeled for you: nuclear membrane, cell wall, and cell membrane. And then it says which features are also found in prokaryotes. So also prokaryotes was only X and Y. There is no nuclear membrane in prokaryotes. Prokaryotes we mean are bacteria. So prokaryotes are bacteria, and that is something that we all need to remember. Do not confuse this. Bacteria, prokaryotes. Blood pressure, BP. Which of the structures are found in photosynthetic prokaryotes? Now, you know prokaryotes do not have any photosynthetic prokaryotes. So photosynthetic means that they would have some sort of way by which they can convert light energy to chemical energy. But of course, the answer to that is only 1 and 3. Why? Because cell membrane and ribosomes. Because there's no cellulose wall. It is a peptidoglycan wall. And there can never be any chloroplast. There could be some sort of structures which resemble thylakoids, but not any membrane-bound organelle. So please remember that. Then coming on to question 6. In order to estimate the quantity of glucose in a solution, equal volumes of a range of known concentrations were mixed with equal excess volumes of Benedict solution and placed in a thermostatically controlled water bath at 90 degrees Celsius for the same length of time. The unknown solution was then treated in the same way and the colors of the known and the unknown solutions compared. What is the independent variable in this procedure? I've always told you this when I've revised paper 5 with you as well. Independent variable is I, which you have changed. So the concentration of glucose you changed, so that would be the independent variable in this investigation. And then of course you were checking the concentration of glucose. So what was the result was the unknown was the color of the known and the unknown solutions were compared. So basically the colors was the dependent variable. Then coming on to question 7. Now question 7 is what cannot occur as a result of condensation reaction. Now we've done this in a previous paper. Condensation reactions are those in which water exits. So the water molecule, you can see this one here. So if you remember that condensation, removal of water. So basically a bond has to be, which cannot, condensation is the removal of water, but it says what cannot occur. So bond is actually formed. A glycosidic bond will be formed. So what cannot occur as a result of condensation is breakage, because it's usually the bond that is formed during condensation reactions. Then we come on to question 8. Now if you look at question 8, which pair of monosaccharides form sucrose? Now we all know that which pair of monosaccharides. And if you look at the sucrose molecule, this is the sucrose molecule. So it's made of glucose and fructose. So you have to know the chemistry of it and you have to be very clear about it. So which pair, glycosidic bond? So it had to be C. Coming to question 9, the diagram shows the relationship between different polysaccharides and the glycosidic bonds formed between the monomers. And which row is correct? So it was amylopectin, amylose, cellulose, and glycogen. Of course, you know cellulose is beta glucose. So remember BC, before Christ. So beta glucose is always cellulose. So here we've got cellulose, we've got beta, but then of course you see 1 and 3. So you have to see 1 and 3 and 2 was common. So you have to be very careful when you have to know this. If you know the whole of the biology and the chemistry behind it. So 1, 2, 3, 4. So the answer was D. Why? Because 1 is glycogen. 1 is glycogen and 2 is the alpha 1,6 bonds and 3 is amylopectin. So glycogen and amylopectin both have alpha 1,6. And amylopectin and amylose have alpha 1,4. So that is why this was 3 was amylopectin and amylose has alpha 1,4. So you need to revise this to be able to remember this. So amylose has only 1,4, amylopectin has 1,4 and 1,6, glycogen has 1,4 and 1,6. But you can see there are more 1,6 in glycogen as compared to amylopectin. So amylopectin is branched and a glycogen is branched. Now coming on to question number 10. The diagram shows some of the types of bond in fatty acids. Which row shows the bonds found in each type of molecule? So unsaturated fatty acid and saturated fatty acids. So answer is 1, 2, 3, and saturated fatty acids had 1 and 3. So this is one we need to revise the chemistry of fatty acids, and that would make us, this question would make it easier. Then coming on to question 11, what could describe the tertiary structure of a protein? Now the tertiary structure of protein and the options that they've given you is alpha helix, a globular structure, the specific order of amino acids, and a specific three-dimensional 3D shape? Now, of course, the answer was 2 and 4. Why? Because alpha helix is part of the secondary structure, and the peptide bonds are the primary, the specific order is the primary structure. So that is why this was wrong and this was wrong, and the answer was 2 and 4 only. Because order of amino acid is the primary structure. Secondary structure is the hydrogen bonds and alpha helix and the beta pleated sheet. Then we come on to question 12. Following a heart attack, the enzyme lactate dehydrogenase leaks into the blood plasma from damaged heart muscle. So we have this enzyme, lactate dehydrogenase. So they're giving you some information. You need to decipher this information. You need to figure this out. Leaks into the blood plasma from the heart muscle, damaged heart muscle. Which steps are required to obtain an estimate of lactate dehydrogenase? So we want to find out how many of these enzymes are in the plasma. Are there 10? Are they 100? Are they 1,000? Activity in a sample of blood plasma. So what do? You sterilize the blood plasma by heating. Why would you do that? Incubate with substrate for lactate dehydrogenase. Naturally, if you incubate with a substrate, then you would know how much is the product being formed. So if there are 10 enzymes, or if there are 100, or if there are 1,000, so that would give you an idea. And the last one was incubate lactate with dehydrogenase inhibitor. Well, we want to find out, it says, which steps are required to obtain an estimate of the activity, that how much is there lactate dehydrogenase, how much is the heart muscle damaged. So of course the answer was C. You incubate with a substrate, so you know how much product is being formed. So that would tell you that are there 10 active sites, are there 100 active sites, are there 1,000 active sites. Okay, coming to question 13. Two experiments were carried out using an enzyme from humans. The first experiment, X, was carried out at a constant temperature of 37 degrees Celsius. During the second experiment, the temperature was increased from 37 to 80. All other factors were kept the same. Which graph shows the results? So, first experiment carried at a constant temperature of 37. So it was continuously at 37. And during the second experiment, the temperature was increased from 37 to 80 degrees, and all other factors were kept constant. So you got to understand is product concentration. So the first experiment X is this one, and then the second experiment was kept at was increased from 37 to 80 degrees, and all other factors were kept the same. So naturally the product concentration would level off because the enzyme would get denatured, and if from 37 it went up to, so this is what we had to understand, is that the answer was A to this question. Then we come on to the next question, which is question 14. Now if you look at question 14, which molecules are found at the outer surface of a cell surface membrane? Cholesterol, glycolipids, phospholipids. Now cholesterol is usually inside, so that was the only one which was inside. The rest of the glycolipids and the phospholipids found at the outer surface of a cell surface membrane, and the answer to that was D, glycolipids and phospholipids, which are found on the outer surface as well, and the cholesterol is found on the inside. So cholesterol. Which substances, question 15, which substances can pass directly through cell surface membranes without using a carrier protein or a channel protein? So you know if it's an ion, so calcium and sodium is impossible, so 2 was not the answer. Carbon dioxide is a small molecule, and so is water, which is a small molecule. And usually we say that, the easy mnemonic for that is SUM, small uncharged molecules. So small uncharged molecules can pass directly through the cell surface membranes, and carbon dioxide and water are part of that group of small uncharged molecules, either a small molecule or an uncharged molecule. Then coming on to question 16. If you look at question 16, we says which statements about the proteins in cell surface membranes are correct. They can be involved in active transport and facilitated diffusion. They can be involved in antigen recognition. They have hydrophilic R groups to interact with the inner portion of the membrane. Now, of course, we have which statements about the proteins in cell surface membranes. So the answer was B. Why? Because in the cell are which statements about the proteins in cell surface membranes are correct? The inner portion of the membrane is hydrophobic. It's not even the fatty acids are all hydrophobic. So that's why 3 was incorrect. And the 1 and 2 is the correct answer to that. Then coming to question 17. Which row is correct for a human cell just before the cell enters prophase of mitosis? Which row is correct for a human cell just before the cell enters prophase of mitosis? So number of chromatase, number of molecules of DNA in the nucleus, spindle present, nuclear envelope present. So human cell has 46 chromosome. When it enters, it will have 92. So this was the answer then. Number of molecules of DNA in the nucleus, 92. And spindle present, no, not as yet, because it's going to enter prophase. And nuclear envelope present, yes. So spindle will start to form as soon as, but this was before the cell enters prophase of mitosis. Okay. Then we go on to the last question, which of course another student has just asked me the other day. Now question 18, which event is part of the mitotic cell cycle? Now at times they do refer to the mitotic cell cycle as a whole, which means the interphase and the mitosis and the, but you see here there were not many options. I mean, I would have struggled with the options because if we say interphase, then DNA replication and cytokinesis, these are all 3 parts of this. So, but it says which one is part of the mitotic cell cycle. Mitotic cell cycle, if you look at only mitosis, is prophase, metaphase, anaphase, and telophase. Well, there were no other options available to me. So that is why I would have said anaphase. But you couldn't have said cytokinesis because you couldn't have said because all these are part of the interphase and the cytokinesis, because in another question in another paper I remember in the mitotic cell cycle they referred to the whole cycle. This whole cycle was referred to as the mitotic cell cycle. So at times Cambridge refers to that, at times it is only talking about mitosis. So I mean you have to be sensible enough to come up with the answer, and the answer was anaphase. Question 19, the diagram shows part of a DNA molecule. Now, which labels represent purines? You know purines are double ring structures. So you had to come up with what represents purines. So 1 was wrong, 3 was wrong, so it had to be 2 and 4. So that is why you have to figure this out. Purines are adenine and guanine. So you have to remember this and you have to make some sort of a mnemonic to remember these. Then we come on to question 20. Now question 20 was a double-stranded DNA molecule was analyzed and 29% of its nucleotide bases are found to be adenine. Which percentage will be cytosine? And that was easy. 29 adenine and thymine. 29 plus 29 will be 58. So remaining is 42. So there has to be 21. Remember A and T. So A and T. So A and T, if that they told you that was 29% were found to be adenine. So if this is 29, then T is 29. So 29 plus 29 is 58. Now 100 minus 58, and you're left with 42. So 42 divided by 2 is equal to 21. So you have cytosine 21 and guanine 21. Now question 21. When a gene mutation occurs, which of the following may be altered, resulting in the production of a non-functional protein? Amino acid sequence, DNA nucleotide sequence, and mRNA nucleotide sequence. It had to be 1, 2, and 3. Why? Because the DNA nucleotide sequence changes, so the mRNA changes, and of course that could result in an amino acid sequence changing, and that would of course be a gene mutation, which would not be a silent mutation, and it would of course result in the effects the production of a non-functional protein. They said that to you in the question. So if you're an A2 student, You know that there are some mutations which are silent mutations. Then we come to question 22. Now, question 22 was simple. You had to do DNA is GCA, mRNA is CGU. So you have to figure this out. So this was the answer to that. Then question 23, which changes in the water potential and the volume of liquid in the phloem occur when carbohydrate is taken out of a sink into a phloem sieve tube element? And this has come in a previous question as well. The lower, and of course, it will increase the volume of liquid. Then we have question 24. A dendron a dendrograph records changes in the diameter of a tree. The diagram shows a dendrograph fixed to a tree. Some results are shown on the chart recording. Dendrograph fixed to a tree. You have the diameter of the tree and you see the diameter. Say, I always say, put some figures on it. So if this was 10, so it is decreasing. 10, 8, 6, something like this. And then this was the time of the day. What explains the diameter changes recorded during the day and night? Now you see adhesion, cohesion, this is a very nice diagram, polar or charged object, and then how it is going to pull. Cohesion is between the molecules and adhesion is with the side of the wall of the vessel. So what explains the diameter changes recorded during the day and night? Adhesion forces decrease during the night. Cohesive tension forces increase during the day. Mass flow of sucrose increases during the night. Root pressure decreases during the day. Now, the other answers were incorrect. Why? Because it says what explains the diameter changes recorded during the day. So, cohesive tension forces increases during the day. So that's why you can see the diameter decreasing. And this diagram very nicely illustrates this. I like this diagram a lot because it illustrates this very beautifully. How it is going to pull it inwards, and that of course result in a decrease in the diameter. And then we come to question 25. Now, if you look at question 25, which description is correct for xylem vessel elements? Now, you remember xylem, phloem, stem, root. So it says the correct answer is contains elongated cells with end walls broken down, located in vascular bundles in the stem and centrally in the roots. So the others were all wrong because it says cell joined to form a tube, pits. Now this is all phloem. Contains cell joint containing cytoplasm cell walls with lignin. Now there's no cytoplasm. Xylem has no cytoplasm in it. That was the wrong bit. And dead elongated cell lignified cell wall with pits at intervals associated with companion cell. Yeah, pits is correct. Yes, xylem has pits. This one is also correct. Uh, but then, of course, companion cells are in the phloem, not in the xylem vessel. So this is what you have to figure out. Why was A wrong? Cell joint pits at the sieve plates. Sieve plates is found in phloem. So this was the wrong part. So you have to see which one was wrong in every part. And then, of course, you knew that C was correct in this part of the question. Then we come on to 20. Yeah, we have 25. I think we'll complete this here and then we'll continue the next video because I know I can't complete this whole paper in the next video. So I'll complete it in the next video. Okay, so that will be all for now and thank you very much.